Hi, I'm Dr. Olusoji Adeomi and I welcome you to this course. Before we begin, I'd like to highlight exactly what I want you to take away from this presentation. By the end of this study session, you should be able to define and use correctly basic key terms and concepts. You should be able to describe the relationship between genes and heredity. You should be able to list and explain the various branches of genetics, and you should be able to state why the study of genes have become so important to us. I'll begin this course with the question, what is genetics? Genetics is defined as the study of heredity and variations of organisms. Heredity, on the other hand, is a process by which a parent passes certain genes on to their children. Heredity describes how some traits are passed from parents to offsprings. The traits are expressed by genes, which are small sections of DNA that code for specific traits. Genetics can traditionally be subdivided into four divisions, and this will be discussed here. The four major branches of genetics are classical genetics, molecular genetics, population genetics, and quantitative genetics. All genetics before the 1970s are classical genetics, which are mainly based on observing phenotype inheritance. It describes how traits are passed from generation to generation. Molecular genetics studies the structure and functions of genes at a molecular level. Population genetics tries to answer questions about gene behavior at population and evolutionary level, while quantitative genetics is a study of the inheritance of traits that show a continuous distribution of phenotypes in a segregating population. These traits that are controlled by many genes also exhibit quantitative inheritance as each gene segregates in a Mendelian fashion. There were three major events in the mid 1800s that led directly to the development of modern genetics. And these main events were in 1959, 1966, and 1871. In 1859, Charles Darwin published The Origin of Species, which describes the theory of evolution by natural selection. This theory requires heredity to work. In 1866, Gregor Mendel published Experiments in Plant Hybridization, which lays out the basic theory of genetics. This was widely ignored until 1900. In 1871, Friedrich isolated nucleic acid from pus cells. There were also major events that occurred in the 28th century. In 1900, there was rediscovery of Mendel's work by Robert Correns, Hugo de Vries, and Erich. In 1902, Archibald Garrod discovered that alkatonuria, which is a human disease, has a genetic basis. In 1904, Gregory Bateson discovered linkage between genes. He also coined the word genetics. And in 1910, Thomas Hunt Morgan proved that genes are located on the chromosomes using Drosophila. In 1918, Fisher began the study of quantitative genetics by partitioning phenotypic variants into a genetic and an environmental component. While in 1926, Hermann Muller showed that X-ray induced mutations. In 1944, Oswald Avery, Colin MacLeod, and Maclean McCarthy showed that DNA can transform bacteria, demonstrating that DNA is the hereditary material. In 1953, James Watson and Francis Crick determined the structure of the DNA molecule, which led directly to the knowledge of how it replicates. And in 1966, Marshall Nirenberg solved the genetic code, showing that three DNA bases code for one amino acid. In 1972, however, Stanley Cohen and Herbert Boyer combined DNA from two different species in vitro and then transformed it into bacterial cells. This was the first DNA cloning. While in the year 2001, the sequence of the entire human genome was announced. Next, we're going to take a look at the basic genetic concepts and terms. And the first that we'll be looking at is the genome. A genome is the full set of genetic information that an organism carries in its DNA. The study of any genome starts with the analysis of chromosomes. And chromosomes are bundles of DNA and protein found in the nuclei of eukaryotic cells. A chromosome, as you can see here, is a very long DNA molecule and associated proteins that carries portions of the hereditary information of an organism. They are located in the DNA. They are composed of DNA and protein that are located within the nucleus of our cells. Chromosomes determine everything from hair color and eye color to sex. DNA stands for deoxyribonucleic acid. DNA stores genetic information in the nucleus. DNA is the genetic blueprint or the recipe for making all living things. Almost every cell in your body contains DNA. And you have 5 trillion cells in your body, and that's a lot of DNA. An allele is an alternative form of a gene that is located at a specific position on a specific chromosomes. Organisms have 2 alleles for each trait. Now, a gene, however, is the basic unit of heredity in a living organism. Genes hold information to build and maintain cells and pass genetic traits to offsprings. Genes, however, are sections or segments of DNA that are carried on the chromosomes, and they determine specific human characteristics. Now, let's consider the concept of dominance and recessiveness in alleles. Now, a dominant allele is one whose traits always show up in the organism when the allele is present. A recessive allele is covered up or masked whenever the dominant allele is present. A trait controlled by a recessive allele will only show up if the organism inherits 2 recessive alleles for the trait. Now, a dominant allele is expressed even when it is paired with a recessive allele, while a recessive allele is only visible when paired with another recessive allele. Next, we're going to look at the concept of homozygosity and heterozygosity. A homozygous trait is made up of 2 of the same alleles, while a heterozygous trait is made of 2 different alleles. Now, a genotype is an individual collection of genes. The term also can refer to 2 alleles inherited for a particular gene. The genotype is expressed when information encoded in the gene's DNA is used to make proteins and RNA molecules. The expression of the gene contributes to the individual's observable traits, which is called phenotype. Now, as you can see from this session, gene study can be seen to be important in our everyday life. In the future, doctors and scientists hope to use our genetic information to diagnose, treat, prevent, and cure many illnesses. Genes are instructions which tell your body how to make all the proteins it needs to survive and grow. By identifying each of these proteins, scientists hope to better understand how the body works and what is happening when it doesn't work properly. They hope this knowledge will eventually lead to more effective medicines and treatments. In this study session, you have learned the definition of genetics along with some keywords. You have also learned the history of genetics as well as the relevance of genes to life. Thank you.